My name is David Tete and I'm working here this summer in Jeff Hartgerink Lab.
And my name is Erica Bakota and I'm a graduate student in Jeff Hartgerink's lab.
Right now we're taking a tour of the lab and Erica will be showing us around and showing us the lab.
So this is the entrance to the lab. We're going to go up and down the aisles and look inside some of the fume hoods and look at some of our equipment. So these are the fume hoods, and this is where we carry out experiments. This is called a rotovap in this hood, and in the other hoods we just have some chemicals.
The machine I'm staring at right now is the synthesizer. That is where the peptides are synthesized in this lab. And in a little while, we'll open the machine for you to see the wells and then the amino acids in vials and then all the other chemicals that are used in synthesizing the peptides. So to my right hand side is the well where the mixing of the amino acids is done to make the peptides. The chemicals are the ones you are seeing with the red rings around it, and in the middle there are the amino acid vials. These are other people working in the lab, Brennan and O'Hay. That's my bench area where I take notes and carry out my experiments. This is the HPLC which we use in purifying the peptides and also characterizing the peptides. And over here to our right is the heavy duty sonicator, which we use in dissolving the gels here in the lab. This is another section of the lab where we have equipment that are used in characterizing the gels that are synthesized. The first one is the CD, and then the other one is the IR. And we use these machines to characterize the peptides that we synthesize here in this lab. We also use microscopes and then a rheometer. Rheometer is used to measure the viscosity of the gels that the peptides are used to make. And that is the machine right there, the rheometer, which is used to measure the viscosity. Okay, right now I'm at my bench trying to make some arginate hydrogels. So I'm folding a piece of paper, to put on the scale. I'll zero the scale and then weigh out my arginate. I'm making arginate gels of different concentrations: 10%, 5%, 3%, and 1%. And then we look at the viscosities of these gels. Definitely they will be of they will have different consistencies. And then we'll use the rheometer to measure their viscosities. It's important to make sure that you don't spill anything in the lab for safety reasons. Am I washing the radial cylinder that I'll be using to measure the deionized water in which I will dissolve my arginate. Arginate is not very soluble in water, so we need to shake it vigorously to dissolve it, or sometimes we might need to use the sonicator to help us dissolve it. it is not very well dissolved. So I'm placing them right now in the sonicator to help dissolve it thoroughly. There they are sitting in the sonicator.
This is a heavy-duty sonicator that we use when the gels aren't very well dissolved to dissolve them a little bit better. You can see the alginate gel here is pretty thick, and so we'd like to measure the viscosity of this gel. So I'm placing it on the plate of our rheometer. I'm wiping away the excess gel so that we can bring the head of the rheometer down onto the gel. And when the machine is in contact with the gel, it can measure the viscosity, and then it will then send that data to the computer like you see here.